hopefully I'm going to see it start recording in terms of time. That looks good. See, there's a pause button there. We're getting audio and video coming through, which is important because what happened last week? We had a fail for Panopto. Now, I don't want to jinx it and start talking about it badly, like maybe it'll freeze when I'm not looking at it. We have had issues with that in the past. Get everything going, then all of a sudden we can start lecturing. By the time we get to the end, you see it's like, oh, it just locked up, and I don't know why. So hopefully it doesn't get started with that. But we did have that issue last week, which is why we don't see lymphatic or immune part 2 on there in our list of Panopto recorded lectures. But to help with that, notice that up here, of course, we have our usual folder, which is the chapter highlights that we always recommend that you watch ahead of time prior to the big lectures. But then I created a separate folder here that has makeup immune lectures. So these ones here are a bit older, and they are also based on the other textbook, but they do cover the same content, right? It's going to be the same lines of defense, etc., cells and molecules that we're talking about that defend us on those levels. So if you want to be reviewing the information that we were talking about last time, you'll find that content in there. The figures may be slightly different, but overall the content's the same. So you do have a backup copy there available to you. Okay? Okay, so hopefully then we will see our immune system part 3 showing up. Look, there it is right there. As we speak, as I speak, as you listen. You can speak too anytime, definitely. So we'll get you there. So I guess leading to that in terms of speaking, do you guys have questions from what we were talking about last time? Yes, Nubia.
This whole, like, how information starts, what causes it, and just the whole
Sequence.
of
Okay, so inflammation itself, if we're thinking about it in terms of organizing and thinking about our lines of defense, right? What line of defense is inflammation going to be? We've only gone through first line of defense and second line of defense so far. So second line of defense, right? First line of defense, external defenses, right? Trying to prevent things from coming in. And when we say things, that could mean just about anything. So remember that in our lines of defense, we're looking at, in this case, external for first line of defense and non-specific. So they're trying to keep out anything that should not be there. Now, if, like you see in this figure here, we have a breach of our external defenses, and now we have a pathogen that's gone internal, then we have our second line of defense, which is going to be internal and non-specific. Okay? So inflammation counts as a second line of defense. And what we're trying to do here is we're trying to bring more of our defenses to our site of infection, to our site of invasion, if you want to consider it that. So with inflammation itself, inflammation is a process where we are opening up the blood vessels in some local area so that we can make these blood vessels a little bit leakier, so that it makes it easier for cells to get out and migrate to the site of infection, etc. So when we're thinking about, okay, we're trying to classify these lines of defense as, okay, is this a molecule? Is it a cell or something like that? Yeah, you might want to consider this one in terms of inflammation. You might want to consider this one more like a molecule. So it's on the level of a molecular defense, where molecules that are either from the infectious agent or molecules that are secreted by our white blood cells that are already in the battle, already in the fray, or even damaged tissues that have become infected and maybe are dying off. Chemicals from any of those different sources can stimulate this sequence of events that we call inflammation, that will cause our blood vessels to dilate, to become bigger, that will cause even things like our capillaries, like we see in this last one, to get leakier. Those individual epithelial cells will kind of separate a little bit, making more intercellular spaces, or at least making them a little bit bigger. So we have fluids and cells that are now moving out of the cardiovascular system and moving into the tissue where that invasion is occurring. Okay? So there's a few different molecules that can lead to inflammation, right? So we talked about, for instance, histamine coming from mast cells as well as other types of white blood cells. And then prostaglandins and different types of kinins that are also going to work to bring in more of these defenses to the local area. So inflammation counts as a second line of defense. There's a few different chemicals, like we said, that can lead to inflammation, and then it's going to allow other types of cells, other second lines of defense, to get to the area to help out in that infectious area or infected particular tissue. Okay? So inflammation is one of our examples of second line of defense. James?
It says function is to trap microbes. Is that they get trapped because of the defensive cells?
Yeah, so that's a good question too. So not only do we have our different cells that can get to the area, like we see here, right, like our phagocytes that are migrating to the area, but in fact, we can have the creation of different types of fibers, like fibroblasts, etc., can create fibers that tend to wall off the area of infection as well. So that way, rather than allowing it to spread more into surrounding tissues, it helps to create a barrier. It's not a solid barrier, it's not a solid wall, but it decreases the amount of kind of lateral spread that we can get in that infected area. So it also helps to trap them in that particular area. Yeah. Okay? Other questions about first line and second line of defense? Yeah, Zach.
So the complement system is a bunch of proteins in a circle.
Mm-hmm.
Is it like a black hole circle where they're just eating them all, or is it a traction circle, like drawing everything to it?
So that's a good question. So light can't escape from this one. Yeah. But really what it is it's not necessarily attracting things into it. The proteins themselves are attractive to each other in order to assemble this membrane attack complex. But when it assembles, it is really just making a big pore, a big hole in the wall of the bacterium or whatever. And once we create that hole, like we said, it's a bit like creating a hole in the side of your tire or something. All of the intracellular material that we see down here is going to start to leak out of our virus-infected cell or a bacterium or something like that, and it's going to cause the cell to explode, basically the cell to lyse. So what we're doing is we're effectively creating a big hole or puncturing the infected cell or the pathogen to get rid of it. So things can leak out of it instead. So this, again, would be another example. Second line of defense, it's internal, and these aren't cells, they're just proteins. But they help to defend us in this particular way. Okay?
This is also an example of non-specific.
Correct. Because it's not targeting a particular bacterium, it's not targeting a particular virus or a virus-infected cell. It works on a variety of different pathogens. Yeah. Yeah? Yeah?
Also, killer cells,
Natural killer cells, yeah?
So I understand that we have white blood cells that help with immunity and they all do their specific job in targeting something, but I'm a little bit confused with the natural killers because we didn't really talk about them in terms of them being part of white blood cells. Where do they come from? And
Yeah.
If you can explain a little bit about them.
Yes.
Where do they reside? Where are they made? All that kind of stuff.
So the natural killer cells, again, are a second line of defense, so it's an internal defense. They're a type of defensive cell that are going around and trying to hunt down individual types of pathogens. Now, the natural killer cells, they are, in terms of their history, they're similar in terms of the developmental line to lymphocytes. So like T cells and B cells, etc. Now, T cells and B cells we're going to talk about, they actually make up the bulk of our third line of defense that we're going to be talking about today. And they have particular ways of defending us. But in terms of where these natural killer cells come from, they're still going to come from like the bone marrow, like we see all of our other cells. But these instead are specialized, kind of, if you want to consider it, kind of evolved lymphocytes that are granular, which is weird. And instead of some of the other ways that we're going to talk about how other types of lymphocytes defend us, these ones actually cause cells to lyse as well. So they have a function that are kind of like the complement proteins. These release proteins that are called perforins that insert themselves into the membrane of a pathogen and poke holes in it and cause that cell to lyse, much like we talked about the big membrane attack complex. So they come from the same place as our other white blood cells. They defend us in a way that's a little bit different than our T cells and our B cells, and they will be effective against a variety of different pathogens, which makes them a non-specific defense as opposed to our other lymphocytes, which are going to be our specific defenses. They, the T cells and B cells themselves, are only going to target very, very specific pathogens, a particular strain of virus or whatever. Natural killer cells can be effective against a variety of different types of pathogens. Yeah.
And they're floating within the blood.
Yep, so they're migrating, they're going around in the blood, they're going around in the tissues as well. So remember, a lot of our defensive cells are going to be in amongst the tissues, because if we just had our different white blood cells inside of the cardiovascular system, then they would only be effective against bloodborne pathogens. We have to get them out on patrol in amongst the tissues and cells of the body. So they're on patrol, much like neutrophils are on patrol, and monocytes or macrophages are on patrol as well. So distributed widely around the body themselves. Is that better a bit? Okay. All right, so this, another example of second line of defense, but this one we would consider to be a cellular example because these are actual cells as opposed to just molecules. Okay? So make sure that you are, if you want, be making flashcards or whatever, categorizing these things so you can keep track of these. Otherwise, when we're going through, it just seems like, okay, there's another something we talked about, there's another something we talked about, etc. You've got to keep that framework in mind, right? Just like we were talking about the heart itself when we were focused on the heart. Then we started talking about arteries or whatever. You want to be looking at these in terms of lines of defense. Is it a cell? Is it a molecule? And how does it defend us as well? You don't want to, it's good to know, this is second line of defense in terms of natural killer cells, but you should have some sort of description about how it kills off pathogens, right? So in this case, they secrete these perforins that poke holes in the pathogen itself. Okay. So, fast forward, if you will, to adaptive or specific defenses. So these count as our third line of defense. So these are going to be internal, but we've graduated now to the specific defenses. So it recognizes specific foreign substances, and it tries to destroy them. Now, of course, destroying them would be best, but we can also immobilize them or neutralize them, and we'll talk about that too. And this is another thing that we've seen as a pattern where the lines of defense that we have, they are often working together. So even though they're going to try to take out that pathogen when they encounter it, they're also often going to be calling in other defenses to aid in our defense of the body itself. So, for instance, these adaptive defenses, some of the chemicals that they will secrete in trying to get rid of pathogens will aid to inflammation. So it will increase the inflammation and bring in other defenses to the site. And when we talked about the complement proteins, we said that antibodies oftentimes can be one of the initial proteins that are inserted or attached to the membrane, and then the other complement proteins are attaching to that and forming that whole membrane attack complex. Okay? So you can see how they work together with some of our other second lines of defense. Okay. So key aspects of the immune, or the third line of defense in terms of the adaptive immune, is that, number one, antigen specific. So what's an antigen again? It's a marker, right? So it's protein or carbohydrate marker that's attached to this foreign cell. Now we do have self-antigens, right, that identify our cells, and then there are foreign antigens which allow us to identify these foreign agents. So they're antigen specific, so they do care exactly what it is. They're systemic, meaning that they're all over the place, and has memory. So we used the example of chickenpox before, saying that this line of defense is supposed to protect you long term, right? Memory's no good if it's just like I remembered it for an hour, right? Just like studying for the exam or something like that. The idea is to retain it some longer period of time. And so these cells are supposed to protect you over a lifetime. So it's interesting to think about. We've mentioned how there are cells in your body that last, maybe days or weeks or months or something like that, like red blood cells. They wear out and they're gone. But there are other cells in the body that are there over your entire lifetime, things like neurons, etc., sometimes cardiac muscle cells, things like that, and T and B lymphocytes. So there are T and B lymphocytes that were created effectively when you were very young, right, when the thymus was up and running and activating T cells, etc., that then are protecting you over your entire lifetime. They have to be there so that if you get exposed to that pathogen again, they are there to remember it, to know what it looks like, to be able to mount a very quick and intense attack against it if you get exposed to it again, right? There's that whole phrase about those that forget the past are doomed to repeat it or whatever. We don't want that with the immune system. That's the whole idea. We have memory, so you don't have to go through all that chicken pox again or all that particular virus infection again, particular flu virus or something. Once you get exposed to it, then you remember it. And that is also the whole background or whole basis for vaccines, right? So vaccines, and we'll talk about this towards the end, right? Vaccines are where you are purposefully introducing a weakened or a dead pathogen to your immune system saying, look at all this. You could get infected with this in the future. You should know what this looks like. You should mount a low-level defense in this so that you guys recognize it. So if you actually get exposed to it later on, you've already got these recognizable memory cells that are going to be saying, hey, we've seen that before. We know how to deal with it. We're going to get rid of it. Right? That's why you have like a chickenpox vaccine now. You talk about flu vaccines, etc. Okay? So we'll talk about that more later on. Okay. Now, with the adaptive defenses, we've already talked about the fact that there are two different types of lymphocytes, or in this case, just B cells and T cells, that are contributing. So when we look at it, the B cells are going to give us a certain type of defense, and this one is called the humoral or the antibody-mediated immunity. So they're helping us out in a certain way. So when we look at it, antibody-mediated immunity, we already know that antibodies are proteins that are floating around in the blood, and so B cells are the cells that actually produce those antibodies that are floating around. And they're going to be specific to particular pathogens that have entered you, or that may enter you in the future. Now when we look at the humoral part, what does that mean? That's where the whole idea of laughter is the best medicine comes from. So the happier you are, the healthier you are at the junction of healthy and happy, right? No. So when we talk about humors in the body, right, we're talking about fluids, right? Aqueous humor, vitreous humor in the eye, etc. All these fluids in the blood and the interstitial fluid, etc. This is where this defense is going to be located. So the antibodies are going to be floating around in our fluids, potentially coming into contact with these pathogens and then having their effect at trying to get rid of the pathogen. Okay? So this is often called humoral immunity because it tends to be in our fluids. Okay, so the second one, like we said, is called the cellular or cell-mediated immunity, and this is from the T lymphocytes instead. And so one of the things that we mentioned early on when we were trying to make analogies here in terms of our defenses is that the B cells often are kind of like archers, right? Where those antibodies that come out are like the arrows that are shot out, and it's the arrow by itself that's doing the damage to the invader, right? Meanwhile, the B cell is going to be residing far away, like in lymph nodes or places like that. Meanwhile, the T cells, when we talk about cellular or cell-mediated immunity, right, these are more like kind of the infantry doing hand-to-hand combat instead. So with the infantry, right, they're going to have whatever, swords and daggers and things like that have to be engaged right at the defender. They have to go up and bind with the defender and attack them close in like that. So this is often called then cellular or cell-mediated immunity. They have to go head-to-head. Okay, so again, one of the things that we want to make sure that we're focusing on is these three levels of defense. And this now is our third level of defense. It's internal, to be sure, right? We've gotten through the other two. And now this is the specific. It's also called adaptive. And remember that we call it adaptive because it changes through time. It physiologically adapts. It changes. It grows. It gets smarter, and it remembers. Okay? Okay. So one of the things that we need to remember then are these things that are called antigens. So these are the substances, like we said, like the markers on your cells as well as foreign cells, that can, if we're talking about foreign antigens, they can mobilize the immune system and provoke what's called an immune response, where there is a whole bunch of cells that go to attack that particular infectious agent. So we're looking to identify these non-self cells, etc., and try to get rid of them, right? So we have self-antigens and we have foreign antigens. And these foreign antigens, we want to make sure that they can produce an immune response. So the very first thing is we have to be able to discern whether it is us or whether it is foreign. And the second thing is we have to be able to produce a response to it. It's not only important to identify and go, hey, that shouldn't be here, and just let it go. We want to make sure that we can react to it as well. Okay? So a lot of these are going to be the attached proteins that we say are attached to cells, but there are some that are carbohydrates like polysaccharides. There are some even nucleic acids and lipids. So there are other organic molecules that can be reactive like this that we can consider an antigen. Now, what we also find is that there are these things that are called haptens, and haptens are like allergens instead. So the deal here is that your immune system is responding to them as if they are an infectious agent, as if they are a pathogen. But in fact, if you look at the list down here, right, haptens are found in things like poison ivy, dander, detergents, cosmetics, even things like peanuts, right? There's food allergies, etc. So these are small particles that are not necessarily antigens. Like we said, they're not necessarily markers of living things, but they are reactive enough that your immune system will try to mount a response against them for whatever reason. So it's an overreaction of our immune system to something that is non-infectious. And that's one thing to remember as well. You know, we keep using this term pathogen, right? So the pathogen is going to be a disease-causing agent, right? So pathophysiology, right, is where you're studying the diseases and disorders, etc. And so pathogens are going to be these infectious agents that can generate disease. So these are going to be things like bacteria and viruses are the most common ones, but there's even fungi and parasites. Those count as well, okay? So you want to make sure that you sort out the terminology. So pathogens have antigens on them, right? So they are antigenic. Yeah?
You said it was an overreaction to what?
So with the haptens, these are overreactions. These are allergic reactions. So these are immune responses to non-infectious agents. Like we said, like things that you see listed at the bottom there, like pet dander or dust or pollen or whatever, right? When pollen gets inside of you, it's not going to reproduce and turn you into a plant or anything like that, right? But your body's responding to it as if it is. Okay. So when we look at it, right, here's our thing that is an antigen. So this is going to be a foreign antigen that we see here, and there are going to be specific parts to it that are called antigenic determinants. And you can see, according to this diagram, they have particular shapes to them, right? It's kind of like when we talked about enzymes a long time ago, right? An enzyme has a very particular shape. It binds with a substrate that has the complementary shape. It fits like a lock and key mechanism. And the antibodies binding to antigens, or even our receptors for our T cells or whatever, binding to these antigenic determinants, binding to these antigens, it has to have the complementary shape. It has to fit it precisely. And when it does, then it's going to be that particular defensive cells or that particular antibody that's going to be effective against that particular infectious agent. Okay? Okay, and we'll certainly continue with that theme as we talk about how these specific defenses identify these foreign agents. But like we mentioned, our own cells certainly have self-antigens as well. And our self-antigens are a larger molecule that's called the MHC molecule, an MHC protein. And MHC stands for major histocompatibility complex, right? So we're talking down at a cell or a tissue level, saying that it is compatible, meaning that we can get along with it. This is our own tissues. These are our own cells that we want to protect. Okay? So MHC molecules are identifying our cells. Now, technically, there are two classes of them, class 1 and class 2. Class 1 MHC proteins are going to be, for the most part, your normal self-antigens. So, for instance, like with my, work ID, this says that I belong here, that I have access to the lab and everything. This identifies me as belonging here, right? So this would be like a class 1 MHC protein for me. It identifies me as belonging here. It says I'm okay. So these are found on virtually all different body cells because, right, if we have these roaming defenses, right, these security agents that are going through the tissues, etc., everybody's got to have an ID. If it doesn't have an ID, there's a chance that it could provoke an immune response and that we could attack it and try to get rid of it. So we want to make sure everybody's badged up. Okay, so we have self-antigens. Now there are certain exceptions to that. There are certain types of cells that don't have class 1 MHC proteins that are found in specialized tissues. But for the most part, we tag everything around the body to make sure it doesn't get attacked. Right? Class 2 MHC proteins, these are found on some of our cells, but they are found on cells that are part of our immune response. So they are things like when we talk about different types of macrophages, etc., like dendritic cells and things like that go around and eat up pathogens. So what they will do, and we'll talk about this in detail coming up, is that they will serve as what's called an antigen-presenting cell. Is that what we have next? It's coming up. Oh, yeah, see it down there. So antigen-presenting cells, these big macrophages will go up and they will ingest a pathogen through phagocytosis. We saw those steps before. And what they do is they take those foreign antigens that were on the cell surface of that pathogen, and they take them and they put them on their own cell surface. Okay? So what they do is they take their own self-antigen and they combine it with the foreign antigen, and they put that kind of Frankenstein-like antigen on the surface of their own cell. And so the reason that they do that is they're helping to raise the alarm and show our other defenses that we've been infected with this particular foreign agent. Okay? So these class 2 MHC molecules that are on antigen-presenting cells, what they do is they're saying, okay, I'm on your team. Here's my self-antigen, right? I belong here. Don't attack me. But this is what I just ate. And so you should be on alert because I ate this. That means we're infected with it. So you need to watch out for this particular thing. So it combines the two, the self-antigen with the foreign antigen of the thing that it just ate, in a special what's called class 2 MHC molecule that it can then show or present to other defensive cells to let them know that that's something that we need to watch out for. Okay? So a class 2 MHC molecule, like we saw here, is only found on certain cells of the immune response, these antigen-presenting cells. It's a self-antigen, but it has this foreign antigen mixed with it. So it's a little bit like a wanted poster, showing it around to our other defensive cells, saying, this is something that I just ate that you need to be aware of. Okay? Okay. So when we're looking at the adaptive immune system and what are the cells involved and what are the molecules, etc., definitely antigens and antibodies count as major molecules as part of this third line of defense, and we're going to continue talking about those. But in terms of cells that contribute to this third line of defense, B lymphocytes and T lymphocytes and then these antigen-presenting cells are all going to play key roles in defending us on this very, very specific internal level. Okay? Okay. So we know from when we were talking about blood, right? Lymphocytes come from the bone marrow, like all of our other types of blood cells. But ultimately then, they end up developing and maturing in two different sites. So the B cells end up maturing in the bone marrow, and luckily, these letters kind of line up, okay? The T cells end up maturing or going to boot camp in the thymus. Remember, they introduced the thymus as one of the few immune system or lymphatic system organs. So the thymus is that kind of triangular looking organ that sits just superior to the heart. It's bigger when you are born. It gets bigger still when you're in the dirt-eating toddler phase. And then as you reach teenage years, etc., and you've been exposed to things for a long time when you were a kid, then that boot camp, that training ground, we don't need it as much because we've already been training T lymphocytes all the time for years. Now it starts to shrink and shrink and shrink. So as an adult, it gets smaller and smaller and smaller. It gets replaced by connective tissue and fat, etc. Okay? Okay. So T cells mature in the thymus, B cells mature in the bone marrow. In truth, that B actually stands for bird, where they first discovered those, but we can just use the fact that bone starts with B to remind us that B cells mature in the bone marrow before they enter circulation. Okay, so in both cases, what we need to do is we need to make sure that these defensive cells can recognize and protect our own cells, and then be reactive to foreign agents. So what we find, you don't need to know the steps of this, but this is an example of the training that goes on in the thymus. So for instance, here's a T cell that we see here, and here's a thymic cell. This is kind of like the drill sergeant that is teaching them how to be a defensive cell. Okay? So you see, for instance, if we have a thymic cell that has a self-antigen, that's one of our antigens. So we want to make sure that a T cell does not react with that. If it reacts with that, okay, so if it doesn't understand that that is a self cell and it actually tries to attack it or something like that, we kill it off, right? So if in boot camp you give one of these new recruits a weapon and he's just going around pointing it at everybody and can't tell the difference between his comrades and who he's supposed to attack, then we don't want that person in our defenses, right? So that's part of the process that's going on here in terms of the T cells. So if we can recognize the self-antigen and say, okay, that self, I'm going to leave you alone, then that's fine. We can let this one survive, that type of cell survive. Okay? So what we find is if it recognizes it but does not attack it, then those types of T cells can then continue on and finish maturing. So that's the type of training that's going on in the thymus. Again, you don't need to know, right, the different levels, positive and negative selection, whether it's making it through these trials, etc. But just know that that's the type of training that's going on in the thymus. That's why we have a thymus. Okay, and the B cells, like we said, they develop or mature or become immunocompetent inside of the bone marrow. Make sure that we don't react to different cells there. If we have reactive B cells in the bone marrow, then again, those can be inactivated and removed in the bone marrow before they go into the circulation. So B cells complete their whole developmental process in the bone marrow. Once they get released into the blood, then they've already gone through their training. So both of them come from the red bone marrow. T cells complete their training and maturation in the thymus. B cells hang around in the bone marrow. Ultimately, they go out into the system, and we need to make sure that they can defend us. So often they're located in those lymph nodes, remember? They can be located in the spleen and other lymphoid tissues, as well as be out in the blood vessels and in the tissues themselves. So we want them to be everywhere to make sure that they're on patrol. Okay. So this is kind of just highlighting, again, those three major cells, B cells, T cells, and then the last one are these antigen-presenting cells. We said that they are big phagocytes, so they're engulfing foreign particles. They present those foreign particles, those fragments of antigens, on their own surface so that they can be recognized by some of our other defenses, most notably T cells. Okay? So these are some examples of antigen-presenting cells. Dendritic cells, macrophages, which were once upon a time monocytes, and then even activated B cells. So these are all phagocytic cells that will ingest pathogens and then show those foreign antigens on their cell surface as part of that class 2 MHC molecule as a wanted poster to try to activate our other defenses, most notably other types of T cells that are trying to defend us. Okay? So those are three different types of, again, phagocytes that act as antigen-presenting cells. So they'll be an important part of getting this very specific immune response up and running. All right. So, like we said, this whole idea is that we're going to be able to recognize very specific molecules, very specific pathogens, and destroy these non-self particles. We've got to be able to recognize them by their foreign antigens and bind to them. And we notice that they communicate with one another. so that way they can enhance the overall defense against these foreign antigens. Okay? Now, part of the deal here is that with these antigens, right, these antigens are going to be very, very diverse. There's lots of different things that you can come into contact with over your lifetime, right? So bacteria and viruses and things like that, they change through time. You can actually show evolution in real time using bacterial colonies. They will basically change, right? If you talk about evolution, we're talking about differential survival based on your genes. The genes confer a certain physical characteristic, and if that characteristic increases your survival, then you survive to reproduce and you pass those genes on. That's the whole idea with natural selection and evolution, right? Bacteria, because they have very, very quick turnover time, generation time, they can do that very, very quickly, okay? Remind me, I'll try to put up, there's a video that just came out recently where they showed, they made this huge, huge gel where they had developing bacteria, and what they did is they put these gradations of antibiotics in the gel. So they had kind of a lower level of antibiotic right next to the edges of the gel where they were putting the population of bacteria, and then they had stronger and stronger levels of antibiotic as you reach the inside. And then they let the whole plate and gel grow bacteria. And then they could show the bacteria growing and the populations growing, right? You see all the colonies forming, et cetera. And you'll see them get up to the lines of the antibiotic, and they just wait there. It's a time-lapse video. You see them kind of waiting there, and then you see some of them, again, through natural selection and evolution, make their way into the antibiotic strip. So you see some colonies start to breach that, and then those reproduce, and you see it go into the antibiotic section. And then reach the next stronger section. You see them along the edge there, just kind of waiting and dying and stuff like that. But through all those generations, some of them are going to be adapted to even surviving at that higher level. And you see those colonies start to make it through, and they make it through the next one, et cetera. It's pretty cool. It's kind of scary, but it's pretty cool to see. That's why, right? Did you hear recently, right? FDA banned your antimicrobials in hand soap, right? So for a long time, you're like, oh, I need the antimicrobial hand soap. I can't just have regular hand soap. I'm not going to survive that way. Now the problem is we've been using that for so long that the bacteria are becoming antibiotic resistant or antimicrobial resistant. So those triclosan and some of those additives that they were putting into the hand soap, sure, it was taking out some of the kind of lower level bacteria, but now what you were left with are the hardier bacteria that they just ignore that. And so now they've just recently banned that. So you start looking at that in the store in terms of antibacterial hand soap, you won't see that anymore. So they actually realized the issue that they were generating because of that. Okay, but the point here is when we're talking about antigens, right, there's going to be tons of different bacteria, viruses, and antigens that you are going to interact with throughout your lifetime. And we need to be able to recognize those regardless of whether we've seen them before. And so how are we going to have T cells and B cells that can recognize very specifically these pathogens, right? There are bacteria and viruses that don't exist right now that give it 10 years from now will have evolved and are now moving around the world, et cetera, right? That's one of the reasons where you talk about the flu vaccine and things like that. Right? The flu vaccine isn't 100%. It's not going to protect you because they're basing the mixture of flu viruses, the most common flu viruses in your geographic region. They're creating a concoction of those because these are like the most likely ones you're going to interact with. But you have somebody from Singapore or Australia who gets on a plane with their flu virus and they come over here and you randomly interact with them, you're not going to be protected against that one, right? They can't put everything in there. It'd be a massive shot, et cetera. But when we think about it, right, those populations of viruses and bacteria are going to change through time. And so we have to be ready for anything. So you have to have B and T cells that potentially have receptors on them that can recognize that one particular virus, that one particular bacterium, and then remember it to protect it against in the future. And so, like we said, there are ones that you've never been exposed to yet, and or ones that don't even exist yet that you still have to be prepared to defend yourself against, like 10, 20 years down the road. So the way that we actually generate a diversity of receptors that can recognize all of these different foreign antigens is it's kind of like a whole series of genes, right? It's going to be a genetic basis. Remember that receptors are going to be proteins, right? In order to create those proteins, we are using the rough endoplasmic reticulum and all that cellular machinery inside, and all that comes from genes, right? Remember that the DNA effectively is a big cookbook on, number one, how to make new cells like the original cell, how an epithelial cell can make another epithelial cell as opposed to a neuron or something like that. But number two, it's a big cookbook on how to make all the thousands of proteins that that cell is going to need through time, okay? So a specific gene codes for a particular protein, just like a particular recipe makes a particular dish. And so what we find is that our cells can mix up. Small variations in the genes will produce a whole variety of different types of receptors. And so we have about 25,000 different gene codes that if we subtly change them, all of a sudden we get a different shaped protein, a different shaped receptor that's going to be used. And so it can give us billions and billions of different types of receptors. So what we have circulating around inside of us, we talk about B cells and T cells, but all of those B cells and T cells that you have a slightly different receptor on their surface that has a slightly different shape, okay? So what you could have is, if we have some T cells here that were produced in the bone marrow and got trained in the thymus, etc., on the surface of these cells are going to be receptors, right? These receptors are going to be the way that we're going to identify foreign agents, and these receptors have different shapes to them. So like these particular ones that I've drawn here, we do 3 different shapes to their receptors, okay? And what that means is that if I have an antigen that comes along, like a bacterium or something like that, and we're looking at their surface markers for this, right? These are the antigens. It could be a molecule, it could be a cell with these markers on them, right? This one here, this T cell isn't going to be effective against it. It doesn't have a complementary shape. That one doesn't work well against it. This one, which has a receptor that has a complementary shape for this particular antigen, this is going to be the T cell that will be effective against that foreign agent, okay? So we've got to have billions of different types of T cells and billions of different types of B cells on patrol, ready to identify this particular agent that you've been infected with. And once we identify it, then it's going to be, we need a whole army of that particular one to help get rid of that particular pathogen, okay? So we're going to see that through time. So ultimately then, both the T cells and the B cells, they have, like we said, each one has a different marker type that will be effective against a different pathogen with different antigens attached to it. So we have to go through, it's kind of like, again, like a janitor with a whole bunch of different keys. Got the one lock there, doesn't know which key goes to it. It takes time to go through all these different clones, and this one tries it. Nope, doesn't work. This one tries it. Nope, doesn't work. This one tries it. Oh, yep, that one works. We need a whole bunch of this one then to get rid of that particular infectious agent. And the T cells have markers like that, and the B cells also. That's how we figure out which B cell or which T cell is going to be the one to save us in that particular instance, okay? Now the B cells, when we look at them, they also have, like we said, these different receptors on them. I don't know what particular shape I'm going to make for these. It's going to get a little bit weird here, okay? So with these ones here, okay, the B cell, once it identifies a particular antigen, what it's going to do is it is going to start producing these proteins that we see here, that when they're attached to the cell, they count as a receptor, but once it starts pumping out and releasing these, like arrows, then these, when they detach, are the antibodies that are going to be effective in binding with and attaching to these cells, these pathogen cells, wherever they are, okay? So B cells also have a diversity, like we said, of receptors on them. It's just that these receptors, when they're attached to the cell, we call it a receptor. When it detaches and we start pumping these out into the fluids, these are actually the protein antibodies that are going to help to defend us, okay? Now, when the B cell starts doing that, the B cell graduates and we start calling it a plasma cell. So an activated B cell that's pumping out these antibodies like that is called a plasma cell that you see down at the bottom right there. Okay? So in either case, we have all of these different clones that we have made. We have millions and millions of different types of B cells and T cells, and it's only once a particular foreign agent shows up that one of these different clonal types is going to be selected as the one that's going to defend us in that particular disease. Okay? So it's a bit like, almost like a high school dance or something like that, right? So you've got all these guys like lined up against the wall, and the girl comes on, and she goes along, and she goes, nope, not you, nope, not you, oh, yeah, we seem to be a good match. Let's go do a dance, right? So that type of selection is also occurring in terms of the immune system. So that's called clonal selection. So one particular B cell or T cell that has the correctly shaped receptor to it that then is going to produce our army that's going to defend us against that particular pathogen. So this figure here is much more accurately drawn than my own, and it shows what I was trying to say, right? So if you look here, here's a bunch of B lymphocytes. You can see their receptors on the surface, and here are antigens floating around. It happens to match with this particular clone that we see here that has the complementary shaped receptor. And so what we need is a whole bunch of that one. We don't need these other ones right now. It's only this one that matches up with that particular antigen. Okay? So what we do is we start producing a whole bunch of those B cells. Some of them are going to become these massive, incredible Hulk cells that we call plasma cells, which pump out antibodies. Some of them are actually going to become these things here, which are called memory cells. So the plasma cells are going to war, right? May not survive, etc. The memory cells, those are ones we're going to hold back. Those are the ones that are going to be kept in reserve so that if we get infected with that particular antigen again in the future, it's those memory cells that are going to recognize it and mount a very quick response to it again. Okay? So, like we said, we have to have lots of clones of these so that, number one, we can have these plasma cells to defend us right then, and then we can have these clones of memory cells that have the right receptor shape to be able to protect us in the future. So that happens with, in this example, B cells, but this same type of thing happens with T cells as well. So there are going to be our fighter T cells, and then there's going to be a separate little population that are going to be memory T cells that we hold back for the future. So that gives us our memory part. So it's basically like this, right? Clone army. It's exactly the same. So if you think back to some of the movies for Star Wars, right? The guys might get this or, love it more. The ladies might as well. So if you saw some of those movies, there was some of the second round of movies, right? The newer ones, which are actually the older movies, which messes up your whole timeline, etc., right? There is one of those where they were talking about raising a clone army. And what was the idea there, right? So they go to this planet, they're raising this clone army. You see all these different clones out there that kind of look like stormtroopers, but they're a little bit different, right? So they are a clone army that they're raising. Now that clone army was based off of one individual who's a very effective fighter. Who was that? Jango Fett. Exactly, right? So Jango Fett, Boba Fett's dad, right? Jango Fett was a very effective fighter. So if we need a whole army of that particular one because he's good at defending us, let's make them all like that particular one. So you take those helmets off and they all look like Jango Fett, right? So same thing in terms of your B cells or T cells. If we have one that's very effective against that infectious agent, we need a whole clone army of those ones at that particular time. Each one may get their turn over your lifetime, but right now, it's this clone that we need a whole bunch of. Okay? So if we're talking about T cells or B cells, we're going to make a whole clone army. And the B cells, remember the B cells that become activated, we call them plasma cells. They can secrete those antibodies at the rate of about 2,000 molecules per second. So they are flooding your humors, flooding your liquids, internal liquids, with the antibodies that will then work to defend us, right? A bit like arrows, like we said, that are going to stick to the antigen and work to get rid of it. I mean, we had a demonstration of that at the beginning of the semester when we were talking about blood typing, right? We added antibodies against A, against B, or whatever, and what did it do? It agglutinated. It formed clumps. So rather than letting those foreign red blood cells run all around the sample, we attached them with the antibodies. We hung onto them like molecular handcuffs, and we kept them in one place instead. We know that that's going to be good because we have these other things like macrophages, et cetera, in your defenses that can then come to that area and take care of all of those cells rather than having to track them down individually. Okay? Okay. So, like we said, the clones, whether we're talking T cells or B cells, some of them are going to go into the battle at the particular time, and then some of them are going to be held back and become memory cells. So that way, like we said, through subsequent exposures of the same antigen, we'll be able to defend ourselves later on. Okay? So that's a key aspect to remember, is that we're going to have to fight the battle immediately and get you better, but we also want to be able to remember that particular pathogen. So what we find is with this immunological memory, you have a primary immune response upon first exposure, where that one, it takes some time to get over that because it's the very first time that you've seen it. And we have to go through this whole dance trying different clones out on that particular pathogen until we find the one that fits it. And then once we do that, then we get a whole clone army going, right? So peak level of, like, for instance, antibodies, if we're talking about B cells, take about 10 days. It takes that long to identify it and then get that army up and running. And then after that, we're going to see our antibody levels decline. A secondary immune response is coming from those memory cells where we already have some of these clones that are in reserve that can then recognize that same pathogen and get rid of it much faster. Antibody peak levels in the secondary immune response, 2 to 3 days as opposed to 10 days. So it's a much more vigorous response like you see here. So here's the primary immune response here. This is kind of the concentration of antibodies on the y-axis. This is time on the x-axis. So this little peak that you see here, it takes longer for it to generate. This is first exposure here. It takes longer to generate and get over, but the secondary immune response, which is mitigated by your memory cells, is much, much faster. So you get a much more vigorous response that then peaks very quickly to get rid of that particular pathogen. Jane.
So is there some chemical that's released that calls like all the cells that have the right receptor?
So that's certainly a good question. There are some, like we said, some types of like kinins, cytokines, and things like that that will bring the whole inflammation, stuff like that, will bring our defenses to the area. But in terms of a specific one, a lot of our defenses are random interactions. So it's just like a patrol, right? So if you've seen patrols around campus, right? They're in the golf courses and stuff like that. Well, if you are in the back 40 of campus underneath the bushes there, if they don't go to that area very often, you can, live there and pitch your tent there and not have to go home because you love school so much. You can live there for a long time without being discovered. If you pitch your tent right in the center of campus in the Cienega, they're going to discover you quite quickly. And so that's why our defenses have to be systemic. That's why they need to be everywhere. The more we have, the quicker we're going to interact with them. But a lot of it does tend to be kind of a random interaction. So that's why they need to be everywhere. Okay. So what we find is that, like we said, this third level of defense builds through time. We say that it's called adaptive immunity, and that it has this memory to it because we have two types of cells that we will create. We'll create those clones that go into the battle immediately, and then we create these memory cells. So we're acquiring more and more defenses. Our defenses are getting smarter through time because of these memory cells that are being created. And there's different ways for us to acquire a better developed immune system that you'll hear about, either naturally acquiring it and it gets kind of smarter, if you will, or artificially acquiring it. So for the most part, right, this active, naturally acquired immunity is the way that you would normally think where, okay, I ended up, coming to school or something like that, and I was sitting in class and somebody sneezed right in my face, right? So then all of a sudden, oh, that's pathogen, that's attacking mice, getting past my first level of defenses, etc. So when you come into contact naturally with a pathogen and you have to actively mount a defense against it and spend your energy to develop these cells, these clones, and attack it, etc., that's called active naturally acquired immunity, right? So it's just a natural type of sequence of events. You actively had to expend energy in order to defend yourself against it. There are also what are called naturally acquired immunity that is passive, right? Passive doesn't require any energy. This is an example of antibodies that are passed between mother and fetus or mother and baby. So these antibodies are naturally produced, right? But they're not produced by the baby. They're produced by the mother. And so it doesn't require any energy from the baby to have these antibodies floating around and helping to defend them. But likewise, it doesn't last a long time because these are produced by the mother, transferred to the baby. So it's naturally acquired, doesn't require any energy. So this is a type of protection that young infants can get naturally. Now we go to the other side over here. Artificially acquired. This means you are purposefully introducing it to your system. This is like your mom telling you to go down the street and play with Billy who has chickenpox. It's kind of like that, kind of. So there's two types of artificially acquired immunity. One is the vaccine, right? So with this one, you purposefully introduce the vaccine, introduce these pathogens to your system. Now they're weakened or dead. So in most cases, right, some cases things go awry, right? So in most cases, you're not going to have ones that can get into you and reproduce. So they're in there so your body can check it out and say, what is that? What is that? And mount a response against it, but they're not reproducing and spreading out, so you're not going to get full-blown sick. You may get, right, low-grade fever or not feel well for maybe a couple days, but you shouldn't get the full-blown disease of whatever that is, right? Mumps, rubella, whatever, okay? So that is an active type of immune response because you have to generate all the cells against that pathogen, but it's artificially acquired. Okay? The other one that we see here, passive artificially acquired immunity, is like if you're going into surgery or something like that where we know some of your defenses are going to be breached, they may well actually give you antibodies. Now again, much like the naturally acquired passive, those antibodies were not produced by your body. It didn't take any energy for you to produce those. So it's called passive, and it's only going to be short-lived because those proteins were put into your system. They're going to circulate around until they kind of break down and are removed, and then that's going to be the end of that particular defense for you. So it's active if your cells are recognizing and producing those antibodies and producing those defenses. It's passive if they've been given to you, and then that just defends you for a short period of time instead. Okay? But in any case, what's happening here is your immune system, your memory is constantly getting better. You're seeing these things, you're remembering these things, and you're being able to defend against them in the future. Okay? Okay. Now when we look at these defenses, and we're talking about like B cells and how they produce antibodies, antibodies, like we said, are just proteins. So how the heck does a molecule, a protein that's not even living, how does that defend you? How does that actually get rid of infectious agents? Well, we did talk about one example that you see there with the red blood cells, right? Agglutination, molecular handcuffs, just holding those foreign cells in place is helpful in terms of trying to get rid of them. So that's one mechanism of how antibodies protect you. There's also what's called neutralization. So neutralization, if you have viruses or even like bacterial toxins that are secreted, those antibodies can bind to it and kind of surround it and protect your cells from those different toxins or those viruses, protect them from getting affected by them. So they neutralize the threat. They attack it, they surround it, and now you're protected. Okay? Precipitation is kind of like agglutination and kind of like neutralization. It binds to these small particles, these small antigens, and brings them out of solution. So not only does it hold it in place, but it potentially drops it out of solution as particulate matter that then macrophages can pick up. So it creates a bigger clump of molecules that are more recognizable and easier for a macrophage to eat. We also said antibodies can contribute to the complement system as well, forming this membrane attack complex that creates a big channel into an infectious agent, causing it to lyse. Okay? So these are all ways that antibodies, just as a protein molecule, can help to defend us. And like we said, it's not that they necessarily take out that particular pathogen or pathogenic agent themselves, but what they do is they will help to neutralize it or localize it so that our phagocytes can come in, or we can draw other defenses to it, or in some cases they contribute to the cell itself dying off, right? Causing that cell to explode and die off. So even though they're just proteins, these are the ways that a third line of defense molecule can help to defend us. Okay? So antibodies, third line of defense, these are specific ways that you should know that antibodies help to get rid of pathogens. So don't just say antibodies produced by B cells, they just kill off things, right? I mean, that's good, that you'll get points for that to be sure, but I can't give you total points unless I can tell you know what antibodies do, that they bind to foreign antigens and they can produce these types of effects. Okay? So these antibodies are what we were talking about before. Remember when we were talking about blood and we said in the plasma there's different types of plasma proteins? We said that there's albumin, we said that there's fibrinogen, and we said that there are immunoglobulins. So these immunoglobulins are those antibodies that are floating around. Now there's different classes of antibodies. The antibodies have different shapes to them. It doesn't really matter too much when we look at them. These ones here, the IgG stands for immunoglobulin, and then there's different types. It doesn't really matter too much in terms of the exam. I'm not going to ask you, what are the most common types of immunoglobulins around, what are their shape, etc. But if you look at them, you can see that even these antibodies are found in different places and defend you in different locations. So IgGs are the most abundant ones that we can find in the blood, but you can see how they're found in other areas where we might be infected. IgA, these are the ones that are in a lot of your secretions. So they help to protect our outer layer, so to make our skin a little bit more defensive against microbes, etc. These ones, under stressful situations where you're allocating resources towards just maintaining yourself, keeping awake and studying and all that type of thing, these IgA levels actually go down because your body doesn't have the energy to crank out all these antibodies through time. So that's why oftentimes, right, you have a big exam, you study, and not sleeping, you get through the exam, and then you get to the weekend and boom, you get sick, right? Because you've been allocating all of your resources towards studying and getting through that stressful period, and these types of defenses are now kind of cut back. There's only so many resources, and so these kind of get cut back. So in any case, like I said, don't worry about the specific types of those. That won't be something we would worry about on the exam. But this one right here just shows you, for an antibody molecule like we've been drawing on the board, those binding pockets that you see in the upper right-hand corners, those places up there are where we actually bind with the foreign agents. Okay? And those are the parts that are variable based on the genes so that we can bind to all kinds of different infectious agents, all different types of pathogens. When it's attached to the B cell, this stem is where it's attached to the B cell, and then those are the binding pockets out there that will actually attach to the foreign agents. So you don't need to know, I don't care about light chain and heavy chain and all that type of thing. Just know that these areas, the antigen binding sites, those are the different shaped areas like we were saying here that will allow the specificity to attack particular infectious agents. That's all. Okay, so I think from what we've been going through so far, we've gotten to a lot of what we've seen for third line of defense. We're still going to be, we've been focusing on a lot of antibodies, and antibodies between our B and T cells are which? Antibodies come from which? From B cells. So far on our third line of defense, we've been talking a lot about B cells and antibodies, etc. So we've been talking about our archers. We need to talk about our other half of our third line of defense, and those are the T cells. So we're going to be doing that coming up. So we haven't totally finished talking about antibodies, but I realize It's a lot of dense information here where it's just molecules and cells and stuff like that. So why don't we break there for today? We'll review what we talked about for B cells and antibodies next time, finish that up, and then we're going to be moving to T cells very soon. Okay?